Welcome everyone to the Brain Space Initiative. It's my pleasure to introduce Dr. Archana Venkatraman, who's currently an associate professor in electrical and computer engineering at Boston University. She previously was at Johns Hopkins University and last year had moved up to Boston. She currently directs the Neural Systems Analysis Lab, and she's affiliated with biostatistics and biomedical engineering, as well as several other centers. And so she's got some really interesting work she's going to share with us today, looking at biologically inspired deep learning. Very much looking forward to your talk, and thank you for joining us today. I'll let you take it from here, Archana.
Okay, thank you. Thank you, Vince, for the introduction, and Armin for the invitation. So it's a pleasure to be speaking to you today at the Brain Space Initiative. So diving right in, today I'm going to present a brief overview of my research program and then highlight two projects that blend scientific insights with AI for biomedical imaging and informatics. So to set the stage, here are some core statistics. So by the end of this year, about 1.3 million children in the United States will be living with autism. Over 50,000 young adults will develop their first schizophrenic break. There'll be about 800,000 incidents of stroke. More than 18 million people will go through a major depressive episode, and over 6 million elderly adults will start to show the early signs of Alzheimer's disease. So the take home here is that neurological and psychiatric disorders have devastating effects and impacts across the lifespan, but they're also very complex. And in fact, we can look at each disorder or phenotype through multiple lenses. So the most obvious lenses are brain structure, or how regions of the brain are organized and wired together, and brain function, or how regions of the brain activate and communicate with one another. But there are other factors at play too, like genetics, which we'll get to in a bit, the environment, so cues, triggers, the supportiveness of the infrastructure, etc., and other aspects of physiology. So my research lies at the intersection of biomedical signal and imaging data, artificial intelligence, and clinical neuroscience. So effectively, my lab develops new computational models and algorithms that tries to harness the information in these various modalities for targeted biomarker discovery, therapeutic planning, and outcome assessment across a range of neurological and psychiatric disorders. And our long-term vision is really how to fuse these complementary perspectives of the brain and body to better understand and potentially treat these disorders. Now, deep learning has emerged as a very powerful tool to parse complex phenomena and integrate different types of data. However, neuroimaging and just clinical neuroscience in general is a field with unique challenges, and it really prevents a straightforward application of these models. So some of the challenges are unavoidably small and often incomplete data sets. This can be due to the acquisition cost, patient fatigue, the difficulty in recruiting certain sets of patients for the study, etc. Another challenge is massive patient heterogeneity, and that kind of complicates the underlying data distribution that we'd like to model. Another aspect is the need for interpretability, which in some cases is often more important than quantitative performance. And then finally, we tend to have weakly labeled and very messy data. So for example, a clinician might be able to diagnose a test subject as having a disease or not, but we certainly don't have ground truth information about the underlying neural or biological phenomenon to train on. So my lab tackles these challenges using a variety of different modeling techniques. So one of them is formulating deep generative hybrid models. So here the generative or classical component allows us to extract interpretable and actionable information, particularly on smaller data sets, whereas the deep portion allows us to learn nonlinear and complex and convoluted patterns from the data. Another strategy is to embed biological and clinical hypotheses about the phenomenon of interest directly into the model. So this information can be derived from the topological structure of the data that we might be studying, insights from our collaborators, or even the scientific literature. And finally, we've developed new training strategies geared towards learning from weak labels and missing data. So application-wise, my lab works across what I like to call the scientific spectrum, from addressing foundational neuroscientific questions to more translational applications of biomedical signal and imaging data to exploratory directions for understanding cognition and the brain-body interaction. So for the rest of this talk, I'm actually going to highlight two ongoing projects that tackle the domain challenges that I mentioned in different ways. And I'll start with our work on imaging genetics. So as a preview, our strategy in this project is to kind of use a priori information and hypotheses from the literature to design our deep network architectures and models. So the starting point of this particular project is to note that neuropsychiatric disorders like autism and schizophrenia and major depression, they tend to have two complementary perspectives. So on one hand, they're associated with behavioral and cognitive deficits, such as impaired social responsiveness or memory issues. And these deficits can be probed and manifest through altered neural functioning, which we can probe through neuroimaging data. On the other hand, these disorders also tend to run in families, meaning that they're heritable. And so that suggests that there's some genetic underpinning that influences the downstream biological pathways and eventually leading to the observed phenotype. So imaging genetics is an emerging field that tries to link these complementary perspectives and viewpoints to identify more robust and targeted biomarkers. So before I dive into the actual model, I'm just going to provide a very quick overview of the data that we have to capture these effects and sort of the input features to our model. And so my apologies if this is sort of background information for the audience, but I just want to set the stage for how we approach the modeling later on. So functional MRI, or fMRI, is used to measure the brain activity. So it's essentially a 4D acquisition, so it has a relatively high spatial resolution at the millimeter scale, but a very slow sampling rate, so between 2 and 5 seconds, which is much, much slower than neural activity. So as we can see in the figure, we can think of fMRI as a collection of brain volumes over time, or alternatively, each spatial location has an associated time course. So many fMRI studies rely on a carefully designed paradigm to activate specific neural processes. So for example, if we're interested in visual response, the subject might see a series of images in the scanner. And if we're interested in a motor response, we might do a finger tapping task. Now, once acquired, the fMRI data is analyzed using a multivariate regression framework known as a GLM, or general linear model. And at a very, very high level, we're going to model the time series for each voxel as the product of a design matrix that encodes the stimulus, or the paradigm timing information, and then a vector of activation coefficients, which we estimate from the data. We can then use these activation coefficients to construct a contrast map of relative changes in brain activity in response to the stimuli. And this contrast can be aggregated across different brain regions of a given parcellation, and that produces an imaging feature vector, which we're actually going to use as input to our models. Now, in contrast to fMRI, genetic variation is most often quantified using signal nucleotide polymorphisms, or SNPs. So SNPs are a substitution at a particular loci in the genome. So for example, if most people have a CG pair at a certain loci, then a SNP would correspond to having an AT pair there instead. And so SNPs are encoded categorically for downstream analysis, where 0 indicates no change from the most common type, 1 denotes a change on a single chromosome, and 2 denotes a change on both chromosomes. And the most common analysis for SNPs is called a GWAS, or a genome-wide association study. So at a high level, we use a very, very large sample, so think tens of thousands of subjects, to compute the relative, or compare the relative frequency of each SNP between a group of patients and a group of controls. And this gives us a p-value for the genetic association between each SNP and the disease. And so in most studies, we might use a stringent p-value threshold to extract a fairly reduced set of SNPs. So going from over 100,000 in the original analysis to maybe on the order of 100 statistically significant SNPs. And then similar to fMRI, these sub-selected SNPs can be organized into a genetic feature vector. Now, there are a few broad categories of imaging genetics methods. The first is a linear regression framework. And so here the goal is to map that genetic feature vector onto a single imaging feature vector, or a single imaging feature. So while this approach has been one of the most popular techniques in the literature, it provides a limited perspective of neural functioning, since effectively we're constructing a regression between our vector of SNPs and a single brain imaging feature. And in addition, in most cases, the phenotype is not factored into the model. So the identified relationships might or might not be discriminative of disease status. The second method is almost like an alignment technique. So here the idea is to learn a projection that aligns the imaging and genetics feature vectors. So common methods here might be canonical correlation analysis or parallel ICA. And while this approach provides more information than a single regression, it relies on a pair of modalities, which cannot really capture multiple snapshots of the brain or a variety of data that you might want to aggregate. And once again, often the association with the disease is post hoc, so it's not really used to guide that alignment procedure and might or might not be informative of disease status. So there are a couple of methods in the literature that focus on combining imaging, genetics, and diagnosis. So the first is a Bayesian model, which was proposed by Batmangle et al. Several years ago, which used a parallel logistic regression framework to first map the SNP data onto the brain neuroimaging features and then the neuroimaging features onto diagnosis. And so while this was a seminal contribution, the approach cannot recover direct genotype disease relationships, so it's kind of this multi-stage pathway, and it also requires and relies on a single imaging modality and can't really capture different aspects of the brain structure or function. The second approach is some of our early work, which I won't get into too much in this talk, and which used a couple dictionary learning models to project the imaging and genetics data into a shared low-dimensional space, analogous to a CCA, but we also enforced that that low-dimensional space should be predictive of disease status. And so that allowed us to identify kind of robust pathways in both data sets that were also discriminative. So while we were able to demonstrate improved phenotypic prediction, it again relied on kind of the SNPs and a single imaging modality. And finally, I just want to point out all of the methods that I've described rely on the reduced genetic feature set. So the inputs to these models were on the order of maybe 100 to 1,000 significant or highly significant SNPs, and that might throw away a lot of information about genetic interactions and genetic pathways that we'd essentially like to capture. So in our current work, we're using deep learning to overcome these challenges. Our overall framework is similar to our couple dictionary learning framework, but essentially now all our operations will be performed with neural networks. So this work was done by my former graduate student, Cheyenne Gosal at Johns Hopkins, and our collaborator, Anand Mate, who was formerly at the Lieber Institute for Brain Development. So the secret sauce of our framework is really in how we encode the data. And this is how we're able to provide interpretability and incorporate a priori information. So starting with the imaging, as I described earlier, we have an fMRI feature vector where each element corresponds to a brain region. So for example, we might have a parcellation of 246 regions based on the Brain Tome Atlas, but you can also sub in your favorite parcellation here instead. Now, our hypothesis is that only a subset of brain regions are informative for the downstream data fusion, meaning associated with the SNP information and also disease diagnosis. And so this is analogous to a sparsity prior. And we're actually going to automatically learn which regions are involved, so that kind of sparse set, using dropout. So here, Z is a stochastic dropout layer where the irrelevant regions are more likely to be masked, so those black or dark gray boxes, and the important regions are more likely to be preserved in sort of feed forward through the network, so a lighter gray to white boxes. And so in order to learn this importance map in a traditional optimization, we're going to model this mask as a continuous relaxation of a Bernoulli random variable. where the vector P encodes the feature selection probabilities that we can learn through the optimization of the deep network. And then U is a uniform random variable that we sample and add to every feed-forward pass through the network as stochasticity to avoid being trapped in local minima. So following the learnable dropout layer, we use a simple cascade of fully connected layers to obtain the latent encoding. And going back to the model overview, we can use the same encoding structure for any number of imaging modalities. So in the particular results that I'll highlight, we have two fMRI paradigms, which correspond to two branches in the encoder. And then finally, the decoders are almost like a mirror image. They're just simple up projections using fully connected layers. And this takes us to the question of how do we encode the genetics data? And here's where we're going to draw inspiration from the biology literature. So if we step back, we can recognize that the SNPs, so these isolated substitutions, don't actually work in isolation. So instead, they either lie within or are proximal to different genes. And these genes also do not work in isolation. So instead, they contribute to complex biological processes. So our strategy is to design our encoder to mimic a hierarchical ontology of biological processes. And this is something that's been established in the biology literature that we can actually go and look up. And so an example pathway through the ontology is shown on the slide. So we start at more specialized processes like neurogenesis, and it becomes more generalized as it goes up the hierarchy. Now, if we take a step back, we can note that this ontology is very representative of a graph where each biological process or term is like a node, and then the links between them are like edges. And so that's going to give us the structure of our neural network. So in terms of our encoding, our first step is to group the SNPs according to the nearest gene and aggregate that information to a gene score. So this is actually a standard procedure in computational biology based on a weighted summation of genetic risk from a sort of a separate standalone GWAS study. It's actually not a deep learning operation, and it's the only non-deep learning portion of our model. So now once we have these gene scores, those blue triangles, they're going to act as input to our deep neural network. And we're going to learn a projection of the gene scores onto the biological processes, or the green boxes specified by the ontology. And so these processes are going to correspond to hidden nodes within the deep neural network. And the projection operation is analogous to a sparse perceptron layer, where only a subset of the input nodes are connected to each hidden node. And then to sort of simulate information flow, we're going to use graph convolutions to model this information propagation between the biological processes, once again, as governed by this ontology. And as an added layer of interpretability, we actually learn an attention weight on each edge that captures the most prominent pathways of information flow. And in fact, these attention weights are subject specific. So they're based on the encoding structure, or the encoding structure in the latent encodings. And so that allows us to compare information flow within and across cohorts. And finally, we use hierarchical pooling to coarsen the graph at each convolutional stage until we reach a final encoding at the root nodes. So going back to our model overview, our genetic decoding uses a mirrored set of graph deconvolutions based on the same ontology. And our latent fusion is simply a weighted average of all the encodings, where that denominator mn corresponds to the number of modalities present for each subject. So in this figure, if we have both fMRI paradigms and genetics for a particular subject, mn would be 3. But if, for example, the subject did not get one of the fMRI scans due to fatigue or just clinical judgment, that would be 2. And so this strategy allows us to handle missing data because it keeps the latent encoding at the same scale, regardless of the number of modalities. And during training, we can just freeze the missing branches of the network for particular input data while updating the weights in the remaining branches. And finally, our classifier is a simple cascade of fully connected layers. And just as an aside, so we've formulated and I've presented this as a classification task, but we can easily incorporate and adapt this framework to other phenotypes just by changing that final layer. So maybe from a classification to a regression, or even a multivariate regression if we have more sort of output phenotypic variables that we care about. So our loss function during training consists of three terms. There's a reconstruction error for each branch, which provides some regularization. The prediction accuracy, in this case it's binary cross entropy, and the KL divergence term that encourages sparsity in the imaging selection probabilities, so those variables P. So we've validated our framework on a population study of schizophrenia that contains two fMRI paradigms. So here the NBAC paradigm assesses working memory, and the SDMT paradigm queries visual encoding of neutral and aversive stimuli. We use the brain tome atlas to define 246 cortical and subcortical ROIs, and our fMRI feature vectors for each paradigm are the average contrast within each region. Genotyping was done using an Illumina chipset with Plink for preprocessing, and this gave us roughly 100,000 SNPs, which we then grouped to the nearest gene with a 50 kilobase pair overlap. And then the group SNPs were used to create a roughly 14,000 dimensional gene score vector, which was the input to the deep neural networks. And the projection weights were obtained from an auxiliary data set to avoid biasing our model with information leakage. Finally, our data set consists of 1,848 subjects with just SNP data, and that we use to pre-train the ontology network, just using the genetics branch and the classifier. And then we have an additional 210 subjects with both imaging and SNP data. However, many of these subjects completed only one of the two imaging paradigms, so there's considerable missingness in the data. So our first result is to quantify predictive performance, so diagnostic performance of schizophrenia versus control using repeated tenfold cross-validation. And so this table reports the patient versus control testing accuracy across several methods. To parse this a little bit, the first row is a classical approach. So here we use parallel ICA to construct a low dimensional representation and then random forest for classification. And because both PICA and random forest require complete data, we actually need a data imputation strategy to handle the missing modalities, and that might contribute to the uniformly poor performance. Our next two rows use the same autoencoder classifier model, but we replace the ontology network with an ANN with the same number of tunable parameters, so a fairly large ANN. And as seen, regardless of whether we pre-train on the genetics branch on the 1848 subjects or start with a random initialization, our performance tops out at an AUC of about 0.67, 0.68. Similarly, we can also decouple the imaging and genetics branches, this time using the ontology network. And in both cases, again, we see that same top out of 0.67, 0.68. And then finally, our combined imaging genetic framework achieves the best overall performance, and that really underscores the synergy between these two viewpoints. And it also suggests that our biologically inspired deep network can extract these robust and predictive information even on smaller data sets. Now, beyond predictive performance, one of our goals was interpretability. So on the imaging side, we can visualize those feature selection probabilities from the learnable dropout layer. And so for NBAC, we see that the frontal cortex, so the MFG, IFG, plays a key role in memory formation, and it's selected here. And for SDMT, we note the cuneus and areas of the occipital lobe, which play a role in visual processing. We can also do a semantic decoding of these regions using the Neurosynth repository, which aggregates activations and keywords across reported fMRI studies. So this meta-analysis reveals highly overlapping concepts between the feature selection probabilities and the two tasks, but there are also some key differences like vision and action for SDMT and memory retrieval for NBAC. Now, for genetics, we actually can evaluate the information flow across biological processes. So remember, the attention weights are subject specific, so we can sort of look at what that's telling us. So our strategy here, in terms of the post-hoc analysis, is to first trace all paths from leaf to root nodes. So these are full pathways through the network. And then we're going to compare the attention weights between patients and controls to identify statistically different pathways. And then finally, once we identify those statistically significant pathways, all those green blocks in the ontology were associated with terms, so biological process terms. So we can then use, we can do a semantic encoding of these biological processes using a standard NLP tool. So these figure illustrates the sort of data-driven clustering of these these semantic embeddings that we obtain from the NLP tool. And we notice a few clusters, such as calcium ion transport and sensory perception and neural action potential that have been previously studied in schizophrenia. But our network also identifies new processes and pathways, which can maybe be used to either probe current experiments or guide future explorations in the field. And then finally, before wrapping up this project, I just want to emphasize that this imaging genetics framework can be applied broadly to a host of neuropsychiatric disorders. In fact, we're now currently adapting it to study autism and heterogeneity associated with different autistic phenotypes and also differences between male and female presentations. And then beyond the brain, with minor adaptation, it can be applied to sort of problems in other medical domains. So for example, in cardiology and cancer diagnosis, by changing that imaging encoder, moving away from fMRI or feature vectors to maybe a CNN-based encoder for an image, or sort of another type of encoder for studying cancer pathology, we can still use the same overall workflow. So now I'm going to turn to a more translational endeavor involving or looking at epilepsy. So as a preview, the particular work I'm going to highlight in this project is going to blend AI and deep and classical techniques in one of these deep generative hybrid models. So to set the stage, epilepsy is one of the most common neurological disorders. It's characterized by unprovoked and recurrent seizures, which have a devastating impact on quality of life and patient health. Now, while epilepsy is usually treated with medication, between 20 and 40% of patients are considered medically refractory, meaning that their seizures persist despite different combinations of drugs. So alternative therapies for these patients rely on clinicians being able to identify where and when seizures are starting in the brain. So EEG, scalp EEG, is arguably the most important diagnostic tool for epilepsy. It's actually the first and foremost modality used in clinical monitoring and evaluation of epilepsy. And here a patient is admitted to the hospital and EEG electrodes are just attached to their scalp, as you see in the figure. The patient essentially waits in the hospital for several days and they're monitored as they have seizures. And then the data is evaluated after discharge. So what's really interesting in epilepsy is despite decades of research into signal processing and machine learning methods for EEG, clinicians rely almost exclusively on manual inspection of this EEG data. So what I mean is that essentially clinicians will stare at the EEG recordings, such as this, and scroll through time. And so in these recordings, the different EEG channels are stacked vertically and time proceeds on the x-axis. And then they'll try to determine by eye where and when the seizure is starting. And in fact, I like this slide because it actually highlights why automated methods struggle. So if I were to say a seizure is starting somewhere on this slide, most of us as lay people and most algorithms, I would argue, would focus on the most salient features of the data. And we would all be wrong because these salient features, so that really high amplitude or high intensity signal in the middle and these very strange oscillations on the right are actually just artifacts. And often the seizure and the start of the seizure can be very subtle. So in this case, the seizure onset, as determined by clinician notes, corresponds to this rhythmic oscillation in the first channel. And even this onset signature can vary tremendously across patients and even across seizures for the same patient. So it is a challenging problem. Now, with that said, there's been plenty of methods developed for seizure detection from scalp EEG, and most of them follow a standard machine learning pipeline that you see on the slide. So essentially the raw EEG comes in, it's pre-processed, features are selected, and then the features are used to make a determination at each time interval about seizure activity. And I would say the bulk of prior work has really focused on the feature selection component. So namely, how do we extract more sensitive and specific signatures of the EEG? So here many areas have been explored, from time frequency features like STFT to entropy-based features from nonlinear signal processing. And then and to a large extent, the detection of seizure activity is made independently for each time interval. So for example, we might chunk the recording into 1 second increments and classify each 1 second as being seizure versus baseline activity. Now, our strategy is to recognize that the seizure is not an isolated event, but it's rather an evolving process. And so therefore, we're going to model the spatiotemporal propagation of this seizure activity and try to use this information to backtrack the time and location of onset. So our framework will rely on Bayesian models, and this is work done by my former graduate student at Johns Hopkins, Jeff Crayley, and our collaborator, Emily Johnson, at the Epilepsy Center at Johns Hopkins. So we can think of Bayesian models as having two parts. First, there is a latent template, which captures unobservable phenomenon within our problem. But this is also where we can inject a priori hypotheses about the nature of our problem. And so we're going to use this latent template to model the spatiotemporal evolution of seizure activity. Now, the latent template is complemented by observed data, which in our case corresponds to the recorded EEG, that we have. So diving into the details, let's start with the latent template. So our problem is based on the 1020 system for EEG electrode placement. This is the most commonly used configuration for epilepsy monitoring across hospitals nationwide. And it's consists of 19 channels on the scalp, which are shown in these green circles. So on top of this 1020 system, I've added these blue lines, which are going to specify a propagation graph. And the way the graph is constructed is that we connect neighboring and contralateral channels because this captures the most commonly observed spreading patterns in the clinic. So we assume that all channels start in baseline, so it's before a seizure begins, and as time proceeds, at some point a channel will turn on, meaning that it starts to manifest seizure activity, and that's captured by the red electrode in the bottom left. And over time, we expect this seizure activity to spread across the graph and recruit more and more channels to the seizure state. Now, of course, we don't know where or when this spreading pattern will start for a given patient, but we'd like to infer kind of a propagation that looks a bit like this. So formally, we capture this via coupled Markov models, where the state of each channel in the next time step will be influenced by the current states of its neighbors. So if we focus on the gray channel T5 in the lower left, in this case, all of its neighbors are in a baseline state, so we have a low probability of transitioning. However, if some of its neighbors are in a seizure state, it will have a slightly higher probability of transitioning to a seizure at the next time step. And if all of its neighbors are in a seizure state, it will have an even higher probability of transitioning. Now, going back to the model overview, now that we have specified the latent template, we can turn to the likelihood model for the observed data. And as we saw earlier, EEG is very complex. So it has a lot of artifacts that look like seizure and baseline that look like seizure. And so we'd like to be able to disambiguate that. So instead of specifying a parameterized likelihood function like a Gaussian mixture model, we're actually going to use convolutional networks to learn an implicit likelihood. However, we can note that there is one issue here. So the data likelihood is actually the inverse of what we want. So effectively, CNNs, the way they're trained is you put in some EEG and the network will spit out a probability or a classification of seizure or baseline. And so that corresponds to essentially a posterior distribution. And we really want the opposite of that. And so it turns out we can obtain that likelihood factor using Bayes' rule. So namely the likelihood, Py given X, is just the joint distribution, which can be factorized as PY and times the posterior divided by the seizure prior PX. Now, moreover, the PY term is constant across baseline and seizure states, so effectively it cancels out during inference. So all we need is this proportionate term where the numerator is the CNN output and the denominator is the seizure prior. And we can approximate that denominator empirically based on the proportion of seizure windows in the training data set. So combining all the portions together, we derive a variational EM algorithm to infer this spatiotemporal evolution and model parameters. Now we've evaluated this deep generative hybrid approach on 2 clinical EEG datasets. The first is an in-house dataset consisting of 15 patients evaluated at Johns Hopkins. Once again, the patients are monitored over several days, so they have multiple seizures. So we have 90 total seizures across these 15 patients. The second is a publicly available data set from the Boston Children's Hospital, or the CHB data set, and that consists of 24 patients and 185 seizure recordings. We compare our deep generative hybrid with 2 baselines that decouple key components. The first baseline uses the latent propagation model, but replaces the CNN likelihood with a parameterized Gaussian mixture. And the second baseline essentially throws away this latent propagation model and just uses CNNs to classify baseline versus seizure activity. So here's the testing performance from a 5-fold cross-validation. And we can see that our deep generative hybrid almost uniformly outperforms the baselines. And that suggests that we're bringing together the best of both worlds. So the latent template gives us a structure prior to make sense of the noisy EEG, and then the CNNs automatically extract discriminative features from that complex EEG data distribution. Finally, our deep generative hybrid also gives us localization information. So here we have 2 seizures from the testing data set in 2 different patients, and the red corresponds to the inferred posterior seizure activity across time. And as seen, our model is able to capture different spreading patterns in each case, which is always a good sanity check. And in fact, the inferred patterns actually correspond with the seizure onset annotated by clinicians, which is shown on the bottom. Now, moving forward, we've started to focus more and more on the localization aspect, which is actually the more valuable information for clinicians. And our first iteration was to extend the Bayesian model by adding a meta chain that tracks when a seizure starts for the patient. Then this onset, so that patient level onset initializes the spreading pattern that I just described. So that this work appeared in McKay a few years back, and it was the first demonstration of simultaneous seizure detection and localization from scalp EEG. We're also exploring deep architectures that automatically learn this spatiotemporal activity. So this end-to-end seizure activity tracking model uses a combination of convolutional encoders, which are kind of similar to our likelihood, and recurrent models to capture that spatiotemporal evolution. And finally, our second recent work builds off the activity tracking to predict electrode level seizure onsets from global training labels provided by clinicians. So for example, in a typical clinical data set, we might just have a lobe or hemisphere of origin. So how do we use that to get finer information about the likely onset channels? So this model uses a transformer to fuse multi-scale information and an attention model to focus the model on specific times of seizure onsets. And we validated this model on data from 2 different hospitals. And it was the first demonstration of cross-site seizure localization generalization in the literature. Now, before wrapping up, I just wanted to state, even though I focus this talk on imaging, genetics, and epilepsy, another key initiative in my lab is connectomics, or understanding and capitalizing on the brain network organization. So a lot of this work will use resting state fMRI to capture interregional synchrony in the brain. So here, some of the key questions we've been focused on are anomaly detection, so key questions are, can we predict multidimensional clinical phenotypes? So here we've looked at aspects from anomaly detection to non-negative matrix factorization. And then some of the translational work that we've done is, can we use this resting state fMRI to identify key areas of the brain that are useful for pre-surgical planning? So one work has focused on eloquent cortex mapping, so identifying motor and language areas of the brain using resting state fMRI. And then more recent work has actually looked at seizure localization from resting state fMRI, and this is all work in progress. And then finally, just as a one-off, we're also working on signal processing methods, primarily related to speech synthesis and emotion conversion. And so that's been a very interesting blend of traditional signal processing techniques with actually diffeomorphic registration from vision. And then finally, as a plug, we've developed some software. So this is an open source EEG visualizer developed by one of the undergraduates in my lab, Danielle Curry. And it allows you to plot EEG signals, visualize them, but also run deep learning models on the data and overlay those predictions on top of the signals. So they can be used either to generate figures for publications or just try and understand what your model is learning. So before concluding, I'd like to thank my fantastic students without whom this work would not be possible. So everyone underlined on the slide contributed to the work in this presentation. I'd also like to thank my collaborators and our funding sources. So to wrap up, I've described a spectrum of open problems for AI in clinical neuroscience, and in the process, I've highlighted 2 projects that tackle the challenges of sample size and data complexity and completeness in different ways. And I'd say that a lot of this is just the beginning. There's really a universe of exciting problems that will require bridging the worlds of biology, engineering, and neuroscience. And I think these endeavors are really where the field should be moving and is moving, and hopefully will improve the lives of many. So with that, I thank you for your attention, and I'm happy to take any questions.
Sorry, I was muted. Thank you very much, Archana. Do folks have questions? We can either unmute you or you can enter them in the chat. So I will start with one that I see, which is from Cyrus, which says, for seizures and EEG, how well does the model work across different subjects?
So, I mean, I think that's a fantastic question. And I think the answer with many, as with many clinical applications, is it sort of depends on the data set and the subjects. We find that seizure detection tends to work well in many subjects. And then often there'll be individual recordings, seizure recordings in a particular subject that are sort of either corrupted or somehow incorrect. And so I think some of our work right now is looking at, like, how do we reject such noisy recordings? And also, one interesting and challenging thing with seizure detection and with epilepsy in general is a lot of the labels that we have for training are actually noisy. So there's a fair amount of inter-rater variability in terms of demarcating the onset of a seizure. And also, just in sort of clinical context, often seizure will be over-segmented, because the understanding is, as long as you focus attention at a certain point of time, they'll scroll back and forth and identify clear onsets later on in the clinical workflow. So we're also looking at training strategies that kind of mitigate the influence of label noise itself.
Great. We have a question from Brad. Let's see here. Oh, I see. So Sally says, I missed the very beginning, so maybe this was touched on, but do you have any plans to work with simultaneous EEG fMRI?
So I think in the context of epilepsy, that's very challenging to do, simply because fMRI, and especially resting state fMRI, is not within the clinical workflow. So it's actually very difficult to collect the data. So right now we're actually focused independently on EEG and fMRI separately as different information about seizure onset locations, with the idea of trying to fuse that sort of down the road in terms of the analysis. I think it would be really exciting and interesting to look at simultaneous EEG and fMRI, but I think logistically we just have not found that to be possible right now.
Jean Liu is asking about your first project. When you integrate SNPs into genes, how do your different GWAS summary results affect the downstream modeling and prediction performance?
So I think that that's also an excellent question. And the answer is it will, right? So because if you're using the GWAS summary statistics to compute that projection, that will actually influence the downstream task. And so I think what we've tried to do is find GWAS analyses or sort of GWAS summary statistics that are at least well matched to the problem that we're studying. And also kind of larger data sets are very helpful in this regard and making sure that we have essentially a very, very clean initial GWAS. But I do think that that's one of the challenges, especially if you're working in clinical neuroscience, is finding, let's say, an autism GWAS or a schizophrenia GWAS, which is also decoupled from your own data set. Because if you use a GWAS that had your data built in, then there's also information leakage. And so we're working on trying to balance those two.
Hi, Vince.
Yeah, go ahead, Armin.
So Navin have a question, so I'm going to unmute him. He wants to ask his question.
Sure.
If I can. Hi, Navin.
Question, Archana, on explainability, like for tasks like, tracing the path in the network and comparing attention weights, are those custom capabilities, or are you finding support from deep learning explainability tools that are emerging? I mean, it's a hard problem.
No, I agree that it's a hard problem. And I think there are multiple ways of looking at explainability. In the model that I presented and in our work right now, they're custom tools. So it's actually built into the network and sort of the network itself to compute these attention weights. And so that way it's linked to the specific edges, which we can then draw parallel to the biological processes. I think in future it would be interesting to explore some of these more generic AI interpretability techniques. I do worry because it is a very, very large network trained on fairly small samples that without that sort of custom built-in technique, how stable some of those interpretabilities will be.
Of course. Thanks.
There was another question about the connectomic data. In that model, are your functional and structural connectivities considered separate modalities?
So I didn't touch on the specific connectivity model here, but in general, yes, they're considered separate modalities, and so we'll integrate them. Some of the currently our kind of favorite technique is kind of using the structure as a loose regularizer on the function. So not a strong regularizer in terms of masking the functional data, but just a soft encouragement through the optimization procedure itself. But I think this is also an open question. So there are many interesting things we can explore here as well.
Related to your earlier point, how do you deal with stability of the estimates in these models? Like, how are you controlling for that? Like if you run it multiple times, for example.
No, I think that's a great question. So there's a couple of things that we're implicitly doing. So we're actually building a fair amount of structure into our models to try and reduce the overall number of tunable parameters in an intelligent fashion, which hopefully buys us something. But we've also started using like repeated cross-validations for all of our analyses and reporting. So at the very least, we're splitting the data in multiple ways, and we're hoping that somehow that stochasticity will provide at least a more stable estimate of the mean and variance of the performance.
Great. Looks like another one came in here. Yeah, multiple random initializations of the model was what Brad suggested. I think that's consistent with some of what you're doing, but yeah.
But yes, implicitly, because every time we run the cross-validation, we're reinitializing. So yes.
So do you think the model, let's see, two questions. What strategies or methodologies do you recommend for establishing a reliable ground truth? This is for your seizure detection problem. for establishing a reliable ground truth, especially for early stage detection.
So, or should I wait till the second question, or?
Go ahead. You can go
Okay.
ahead.
No, I think that's a fantastic question, and this is one of our frustrations, and a lot of the challenges with epilepsy. I think the only really accepted ground truth is if a patient undergoes a surgical resection and is seizure-free. That's kind of the ground truth. The issue is that every stage of the epilepsy pipeline, you're going to get attrition of subjects because they won't necessarily move forward to the next stage. So some of the ways that we're trying to tackle that is just trying to do multi-stage evaluation. So it will be different subsets of patients, but we might evaluate against clinical notes or clinical annotations as the first stage. Some of them will go on for invasive monitoring, and so that gives us a slightly better evaluation. And then some of them will go on for surgical resection and outcomes, which is in epilepsy considered the ground truth.
Mm-hmm.
Yeah.
Great. In your earlier, I think your first fusion model, you have this kind of masking approach, right? Where you're using the sparsity, you're enforcing sparsity on the Z's. And so one of the things I was curious about is, is that a sort of on or off, or do you allow for partial contributions from SNPs or regions?
That's a great question. Let's see. Try and get back there. It's a soft.
Yeah, there it is.
It's a soft masking. Let's go back to this one. It's a soft masking. So Z is a soft mask. You can, based on that parameter, the temperature parameter, control the sharpness of that transition. But it is continuous so that we can optimize it. If it was discrete, we couldn't back propagate through the network.
Okay. Yeah, one of the things that that we've found useful, not for that piece, but just for fusion in general, is extracting, sort of multiple combinations of, weighted modality networks, for example, right, that interact at the later stage. So kind of, I guess, early versus late fusion. Yeah. So how would you characterize your approach? Is it more of a late fusion approach, or would you
My loose understanding is late fusion, you actually make individual modality predictions and then try to fuse that. Is that
are the parameters for each modality informed by the downstream?
Yes. So all of it is optimized.
It's the training, like you're training the entire model as opposed to one arm and then jumping to the next.
Yes, we are training the entire model. With the genetics, because it's bigger, we pre-train, and we don't use a random weight initialization. We'll pre-train on sort of a different data set that gives us an initialization for that fusion operation, but everything's trained together.
Okay.